Good morning, everyone. I'm Dr. K. Ramya, Professor of Physiology, working in Sri Lakshminarayana Institute of Medical Sciences, Pondicherry, which is affiliated to Bharat Institute of Higher Education and Research, Chennai. Today, we'll be dealing with a physiology lecture topic, which is glomerular filtration rate. Before going into the glomerular filtration rate topic as it, we should know what is glomerular filtration and what are the barriers through which this filtration process is happening. There are three important layers of glomerular filtrations which we should be aware of before going into the process of filtration rate. These filtration barriers they are formed by the mainly by the glomerular capillary membrane and also by the food process. If you see, you can note down here in this picture, it is formed by the endothelial layer of the glomerular capillaries. These things you can note down there are a lot of fenestrations here in this endothelial layer which allows the passage of blood through it and helps in the filtration process. And the next layer is the glomerular basal membrane. See, this is the glomerular basal membrane which is lining the capillary. And finally, it is the podocytes. And podocytes here you can make note down there are simple filters or slits like here, things present here which allow the passage of the filtrate fluid to pass through it. So these layers which we see they are highly specific to the size and the charge of the materials which is passing through it. They will not allow the passage of particles which are specifically more than the size of the slit, and they will not allow the passage of the particles whom are negatively charged. Because they are already negatively charged, they will prefer the negatively charged particles and allow easily the positively charged particles to go through it. That is why proteins are not so commonly filtered through this membrane. So this is the picture which gives you the overall function of the glomerular filtration to you. See, this is the afferent arteriole or the glomerular capillaries through which the blood comes into the glomerular capillaries. These are a mesh of capillaries through which the blood gets filtered and goes out to the afferent arteriole. Once it gets filtered, it enters the Bowman's capsule that is into the renal tubule. The fluid which enters the Bowman's capsule here in this region, we call it as the ultrafiltrate. That is the fluid which is getting filtered through all these three layers and comes out and reaches this area of the tubule is called as ultrafiltrate. That is why this filtration process is also called as ultrafiltration. Generally, this ultrafiltrate is completely free of protein. The reason is that the protein molecules they don't get filtered mainly because of their charges and sometimes due to their sizes. So glomerular filtration, there is the ultrafiltration of the plasma in the glomerulus. This mainly determined by two important factors, which are the filtration coefficient, which we also call it as Kf, and also the pressure gradient or Starling forces. We have learned about Starling forces already in plasma proteins, which is mainly governed by the hydrostatic pressure and the osmotic pressure. Osmotic pressure is contributed by the concentration of plasma proteins in our blood, and hydrostatic mainly by the fluid forces. Uh, in general, we call the hydrostatic pressure as the driving force and osmotic pressure as the retaining or the pulling force because it helps in the retention of fluid inside the blood capillaries. Whereas the hydrostatic pressure, if you see, whenever the hydrostatic pressure is higher, it helps in the filtration of fluid. That is why these two forces helps in the maintenance of Starling's forces. Now we should know how it is getting filtered through the membrane. So as I told you, the first primary factor which is important is the filtration coefficient. This filtration coefficient is again dependent on two important things, which is the capillary permeability. Capillary permeability is nothing but the ability of the capillaries to allow the fluid or a particular substance to pass through it. That is the capillary permeability. So think of a slit-like substance at home you would have seen. When the pores in the capillaries are numerous and the size is bigger, the fluid gets or the substance gets easily passed through it. So that is called as the permeability. The same way here in the membrane also, that permeability is highly important because when the permeability is high, the substance can easily be permeable. That is, the substance can easily pass through the membrane and get filtered. And the next important factor is the size of the capillary bed. Sitting of a smaller material, I mean, it is only about the 2 centimeter diameter. For example, I'm saying it is not about the renal glomerular capillaries. So if you want to pour about 1 liter of solution into it, what happens? It will take some time to get filtered through. But comparatively, if you're going to take a bigger size seed, about 10 centimeter diameter, and pour the same amount of fluid through water, it gets filtered easily because the size of the sieve available for the filtration to occur is bigger and the larger. Okay, the same happens here also. So when the capillary bed size larger, the filtration happens at a higher rate. So in this picture, you can note down. See here, this picture it demonstrates all the slits which are present, the pores through which it is getting passed through. And here, this is the efferent arterioles through which it enters, and these are the glomerular capillaries. So the linings are present in the glomerular capillaries, that is the endothelial layer, basement layer, then comes the cortex. And once it gets filtered, it comes out through the efferent arteriole. So molecular size we have been, and whenever we talk about capillary permeability, depends mainly on the size of the molecule or the particle which is getting filtered, the shape of the molecule which is getting filtered, and the charge of the molecule. When the size of the particle is less than 4 nanometer in size, it gets easily filtered and passes through the slit very easily. And so about 4 to 8 nanometers, you see, it does have difficulty in passing through it, but it depends on the size at the particular moment. But when the size of the particle or the molecule is greater than 8 nanometer, it cannot pass through the filtration barrier at all. So it remains in the capillary and it will not be present in the ultrafiltrate. And next comes the shape of the molecule. So when the shape of the molecule is very elongated, it can easily pass through the slits because it can easily enter it. But glomerular or spherical substance, it cannot go through a smaller slit or a smaller pore. That is why the molecules with elongated shapes pass through the capillaries very easily compared to the glomerular shape. Then comes the charge of the particle, that is the electrostatic charge. Positive ions are filtered very easily, whereas the negative ions are repelled. Because I've told you the barrel itself is negatively charged. So whenever a negatively charged particle approaches the barrier, it repels the substance and it will not allow the filtration of a particular substance through the barrier membrane. That is why the proteins are not getting filtered through the filtration barrier because mainly because of this charges. But what happens in case of glomerular membrane disease, which happens in case of nephrotic syndrome or nephritic syndrome or any other renal diseases? The membrane barrier gets disrupted. In that case, it loses its charges. So what happens? The proteins get filtered. That is why in renal function test, the primary very easy investigation we do to screen initially to check whether there is uh, presence of good function of the membrane by assessing the presence of albumin or microalbumin in the urine, which they call it as microalbuminuria. You would have read about this in the biochemistry. So next we are moving on to the factors or the formula which helps it. Okay. So we have seen about the capillary permeability now and the factors which is dependent. Then, so this is filtration coefficient is K here. So we have PGC, PG, IGC and IT. So this talks about the hydrostatic pressure and the osmotic pressure. So here it is about the Starling sources. See, you can see, I've told you when the hydrostatic pressure in the capillaries are higher, what happens you think? So it aids in the filtration. When the glomerular hydrostatic pressure in the ultrafiltrate is higher, it will oppose the filtration. So this formula can help in finding out that. So you can note down here. See, this is colloidal osmotic pressure. So when you take a net amount, the net filtration pressure comes at around 10 millimeter of about hydrostatic pressure. That means when hydrostatic pressure inside the glomerular capillaries is higher and the formula has been applied, what happens? I've told you hydrostatic pressure is a driving force which helps in the filtration of fluid. So it helps in the filtration, net filtration pressure. So now we'll go into the composition part of the filtrate. So what will not be present in the filtrate, as we have discussed in the previous 3-4 slides, proteins will not be present in the ultrafiltrate. Whereas all the electrolytes, the metabolic waste products, all the other metabolic products, non-natural substances, peptides, amino acids may be present in the ultrafiltrate, and mainly what is present? The fluid component. So now we are moving on to see what is glomerular filtration rate. The rate at which the plasma is filtered by the kidney. Kidney in the sense not the kidney acids, mainly by the glomerular, that is the tubules. Okay. So it is nothing but it is the total volume of ultrafiltrate formed by nephrons of both the kidneys in unit time. So if you take it for a minute, it is about 125 ml per minute. And when it's been converted for a day, it is about 180 per liters per day. So amount of filtrate formed by glomerular filtrating membrane of both the kidneys in unit time. 125 ml per minute or 180 liters per day. There are multiple factors which affect GFR. So if you see, I've given few important things here. They are change in renal blood flow, glomerular capillary hydrostatic pressure, then capsular hydrostatic pressure. Capsular in the sense it is Bowman's capsule. Then we have the oncotic pressure. Why oncotic pressure? I didn't mention separately for the glomerular capillary and Bowman's capsule. Any idea? Because the ultrafiltrate, they won't contain plasma proteins. If there is no plasma proteins in the ultrafiltrate, will there be any oncotic pressure created in the Bowman's capsule? No. That is why I have mentioned anything about the oncotic pressure in the capsule. Then the fifth one is the glomerular capillary permeability. Then effective filtration surface area, size, shape, and electrical charge of the macromolecule. So here, first the renal blood flow. Think of the renal blood flow. Whenever the renal blood flow is increased, the blood flow into the glomerular capillaries are increased, what happens? The blood available for filtration in the unit time is more. So if the blood available to be filtered in the unit time is more, blood getting filtered through the glomerular capillaries and resulting in the formation of ultrafiltrate will also be increased. That is why with increase in the renal blood flow, there will be increase in the GFR. Whereas whenever there is renal vasoconstriction or blood vessels supplying the renal organ is being constricted, what happens? The blood flow to the kidney is decreased. Blood flow through the efferent arterioles will also be decreased. In that case, what happens? The blood available for the filtration will be very lesser. So resulting in decrease in the ultrafiltrate formation, which will finally decrease the GFR. Then we are moving on to the next factor, that is the capillary hydrostatic pressure. So we all know that the hydrostatic pressure is the driving force, that is the filtration force. So whenever the hydrostatic pressure of the glomerular capillaries is higher, what happens? It results in filtration. But on the contrary, whenever the hydrostatic pressure of the glomerular capillaries is lower or lesser, what happens? It won't aid in filtration that much in compared to the previous situation. So resulting in decrease in the GFR. We are moving on to the next factor, that is the permeability of glomerular capillaries. So I've told you, when the permeability is higher, the filtration is low. When the permeability is lower, the filtration is lesser. So GFR decreases. Then comes the hydrostatic pressure of the Bowman's capsule. See, it is in the opposite direction now. So when the hydrostatic pressure in the Bowman's capsule, that is in the ultrafiltrate, is higher, what happens? It will oppose the filtration. So whenever there is increase in hydrostatic pressure of the fluid inside the Bowman's capsule is higher, it decreases GFR. In contrary, whenever that hydrostatic pressure is lower, it will increase the GFR because the opposing force. It is the opposing force. When the opposing force is not so stronger, GFR is increased. When the opposing force is very stronger, the GFR decreases. Then we are moving on to the osmotic pressure in the glomerular capillaries. As all of us know, plasma proteins is a osmotic pressure creating gradient. Okay, so they are osmotically active substances. Whenever they are present in higher concentration, the osmotic pressure is high. That is, the colloidal osmotic pressure is high. Whenever they are present in higher concentration, the retention capacity of the fluid is also higher. So, whenever the osmotic pressure inside the glomerular capillaries is higher, GFR decreases because it retains the fluid inside the capillary. But whenever the osmotic pressure gradient inside the capillary is lesser, what happens? It won't retain the fluid. In that case, the hydrostatic pressure will be higher comparatively. The net filtration pressure will be higher, favoring the hydrostatic pressure, resulting in driving force, aiding in filtration, thereby increasing the ultrafiltrate formation, and finally increase in GFR, that is, increase in glomerular filtration rate. Then the next point, messenger cell relaxation. This is present in the layer. Okay. So whenever it is relaxed, the surface area available for filtration to occur is larger. So when the surface area available for the filtration is larger, what happens? There will be increased filtration, aiding in increased GFR. But whenever the messenger cell contracts, the surface area available for filtration will be decreased. Thereby decreasing the filtration rate, decrease at GFR. Okay. Then these size, weight, shape, all those we have seen already. Anyway, I'll tell you once again. When the weight of the molecule is less than 10,000, it is easily filtered. More than 10,000, it is delayed and decreases GFR. And the shape you have seen, whenever it is elongated and slender, it gets easily filtered through the barrier. And but whenever it is globular, what happens? The filtration is difficult. And neutral and positive substance can easily pass through and can increase GFR, whereas the negatively charged particles are not filtered, thereby decreasing the GFR. So this table gives you an overall view of the factors, very primarily common factors affecting the GFR. So the first one indicates the factors which increases GFR, and this one indicates the factors decreasing the GFR. So this picture will give you an idea about the blood flow and its influence on GFR. See, this is the afferent arteriole on this side, and this is the afferent arteriole. So this is the constriction part. Can you note down? It has been narrowed. So afferent arterioles are the one which carries blood to the glomerular capillaries. So GC, glomerular capillaries. Okay. So when it is constricted, what happens? The blood flowing to the glomerular capillaries are decreased. When the blood flow itself is decreased, there will be decrease in GFR. The same way, we have one other factor which also decreases GFR. That is, see, the afferent arteriole is normal without any constriction, but here you can note down the afferent arteriole is dilated too much. so that the blood leading out of the glomerular capillaries occur at a faster rate because of this dilatation. In that case also, the blood available for the filtration through the membrane will be lesser. So that is decreased GFR. Okay. Then we are moving on to increased GFR. Here you can note down. Here the afferent arterioles are normal, whereas the efferent arterioles are constricted. That means the time available for the blood to get filtered here will be comparatively more. So increases the GFR. And another case, see the dilatation of afferent arterioles, and there's no change in the efferent arterioles. It's because there's dilatation of afferent arterioles, the blood flowing through the efferent arteriole is increased, thereby increasing the blood delivery to the glomerular capillaries. It says increased rate of filtration through the barrier, thereby increasing the GFR. So these two schematic diagrams explain how the blood flow to the efferent arterioles and the efferent arteriole influences GFR. Then these are the common physiological conditions that alter GFR. So in general, we know during pregnancy we have increased blood volumes and because of hormonal changes. So what happens whenever the blood volume is more? There will be more amount of fluid available for the filtration. So GFR is increased. Okay. Then I'm not talking about the arterioles now because we have already seen in the previous slide. So posture, sleep, weather, all this can alter. Okay. Then age. In elderly individuals, there will be decreased GFR because of uh, declining effects of the functions of nephron. Okay. So other things I think you can commonly understand. I mean all this. Then we are moving on to the regulation of GFR. Usually, GFR is regulated by two mechanisms, like one is intrinsic mechanism, other one is extrinsic mechanism. In intrinsic mechanism, we have the myogenic theory and tubular glomerular feedback. And in extrinsic mechanism, it is controlled either by the neural or the hormonal. First, the myogenic mechanism. So this is very common for you. I think you would have known all this in CVS. So whenever there is increase in BP, what happens? There is stretching of blood vessels automatically. So whenever the stretching of blood vessels, the stretch receptors on the blood vessels, that is the blood receptors, they get activated. So there occurs depolarization. So depolarization occurs, what happens? Resulting in vasoconstriction. So vasoconstriction, thereby regulating the blood flow to the glomerular capillaries and maintaining the GFR at a constant rate. And we have one other mechanism through which it is being regulated, that is tubulo-glomerular feedback mechanism. Here, in which it is mainly dependent on the amount of fluid flowing through the tubules. So, whenever there is increased flow of fluid through the tubules, what happens? There will be increased delivery of sodium and chloride in the tubules. So, the macula densa in the juxtaglomerular apparatus will sensitize the concentration of sodium chloride in the fluid filter. So whenever it is increased, what happens? The sodium chloride absorption will be increased, which increases the activity of sodium potassium ATPase. Adenosine and ATP will be produced in excess of the routine, so which acts on the receptors and causes constriction of the efferent arterioles, thereby decreasing the GFR in turn. And apart from this GFR has been auto-regulated, okay, by all the steering mechanisms. Why we have to know all those tubulo-glomerular feedback and myogenic theory? Because these two theories, if you see, they explain the basics of maintenance of GFR in the range of about, they are normal, that is 125 ml per minute, whenever the BP is ranging between 80 to around 180 without any change in the GFR value. Okay, but whenever it is the blood pressure, mean arterial pressure is very lesser than 80 or higher than 180, it gets altered. So this regulatory mechanism to maintain GFR in a constant rate, which is called as auto-regulation, which is carried out by the above two theories which we have discussed. So this graph you can note down. it explains the autoregulatory mechanism. See, here initially when the mean arterial pressure is less than 80, you can note down here because the x-axis explains the mean arterial pressure and the y-axis explains the flow rate. Okay, so till 80 you can note down there is only a gradual steep like increase, but from 80 to 180, even if there is increase in the blood pressure from here to here, you can note down it is maintained at around 125 ml per minute. Okay, then whenever the blood pressure is cross 180, this GFR also increases along with the increase in blood pressure. So mostly the GFR is maintained at a constant rate between 80 to 180 millimeters of mean arterial pressure by this process of autoregulation. Then they are also maintained by neural and hormonal. I've told you. The hormones, major hormones like angiotensin 2. The main stimulus to the angiotensin 2 to come into action is the ECF volume or the blood volume. Whenever it decreases, the blood pressure also decreases. So there is stimulation for the release of angiotensin, which finally results in the constriction of the arterioles. That when afferent arteriole is also constricted, what happens? It decreases the blood available for the filtration in the glomerular capillaries, thereby decreasing GFR. Then we have one other hormone called atrial natriuretic peptide, which gets released from the right atria whenever the atrial wall is stretched out due to increased blood flow or increased PCF volume. So this causes relaxation of mesangial cells. You remember that tabular column which we saw in the previous slides? Whenever the mesangial cell relaxes, the surface area available for the fluid to get filtered will be larger. When the surface area is larger, the filtration occurs at a faster rate, thereby increasing the GFR. So that increases GFR. So GFR can be measured by many modern imaging techniques nowadays, and it can also be measured by the old gold standard methods like renal clearance test using various substances. It can be exogenous or endogenous substances. So renal clearance, if you see, they are nothing but the renal clearance is nothing but the volume of plasma from which the substance is completely cleared per unit time. The formula used for calculating this renal clearance is C is equal to UV by P. We have written CX, UX and PX here because the X indicates the subject which we use. C is the clearance of the substance. U is the urinary concentration of the particular substance. V is the urinary flow rate. And P is the plasma concentration of the subject. In specific, if you see, we use inulin to measure GFR. Okay. The reason or the eligibility criteria for using a particular substance is that the subject should be freely filterable. It should not be reabsorbed and either secreted. It should not be synthesized, destroyed, or stored in the kidney. It should be non-toxic. Concentration in the plasma and urine should be easily measurable for analysis. Okay, and that is why inulin clearance is always used as a high standard and high accurate technique for the measurement of glomerular filtration rate. And we can also use creatinine clearance for measurement of GFR. Okay. So this is a graph which helps in the plotting of the relationship between plasma creatinine and GFR. Then for renal plasma flow, we use the substance specifically called para-aminobutyric acid, which can be injected intravenously. And then by using the formula, we can measure the urinary concentration, plasma concentration, and by applying those values into C is equal to UV by P formula, we can measure the renal plasma flow. So you can now note down that GFR can be measured by inulin and creatinine clearance, and renal plasma flow can be measured by PAH. So these are the three substances in common. See, we have multiple other substances which can be used for assessing the renal clearance. But these three you will have to remember primarily and we'll have a separate session on the measurement of GFR again. To summarize, if you see GFR, it is the glomerular filtration rate, which is defined as the volume of fluid filtered by all the nephrons of both the kidneys in unit time. The normal values are 125 ml per minute and 118 liters per day. The factors affecting, you have to consider always about the blood flow through the afferent and the efferent arterioles. Then the Starling uses hydrostatic pressure and the osmotic pressure, and the net filtration pressure, filtration coefficient, the hormonal influences, and the neural influences. And the surface area, weight, size, and charge of the particle. So take all this as the keywords are the key points with which you will be building up and reading on GFR. Thank you for listening to the class.